Carotenoids are yellow to orange pigments that lend color of certain flowers, fruit, vegetables, corn kernels, fall leaves, salmon, shellfish, egg yolks, and feathers of various birds. Carotenoids are also internally in many organisms. Over 1,100 naturally occurring carotenoids have been described. Most animals cannot produce their own carotenoids. They rely on a dietary supply of carotenoids with roles in essential functions. This video focuses on two carotenoids, zeaxanthin and lutein, and their roles in opposing damage by intense light in both plants and in the human eye, excessive accumulation of radicals and other oxidants that lead to a host of human diseases and disorders, and cognitive dysfunction, damage to extremophile microorganisms in the most inhospitable environments on Earth. Carotenoids are classified as either carotenes or xanthophylls. Their interaction with visible light allows carotenoids, or their derivatives, to serve in human vision and in the sunlight-fueled food production of photosynthesis. The light-absorbing component of the human vision protein is derived from carotenoids like beta-carotene that is converted to vitamin A. Zeaxanthin and lutein also serve as molecular rivets that span biological membranes and lend unique support to membrane stability and function. The xanthophylls zeaxanthin and lutein also protect against damage from too much light in eyes, leaves, and other green parts of plants. The xanthophylls intervene at several stages of a dangerous series of reactions where light-absorbing pigments pass energy to oxygen and cause oxidation of membrane components. This intervention by xanthophylls can: first, safely remove excess excitation energy from light-absorbing pigments before they can pass it to oxygen. Second, detoxify reactive oxygen species. The third, recycle oxidized membrane components. When a carotenoid removes reactive oxygen or recycles oxidized membrane components, the carotenoid itself becomes oxidized and can perpetuate dangerous oxidation cascades. For this reason, carotenoids must be consumed together with additional antioxidants that can recycle the oxidized carotenoid through a series of antioxidant reactions. Carotenoids and vitamin E, both soluble in membranes, support each other in breaking oxidation cycles. Intertwined antioxidation reactions require a mix of essential micronutrients, such as those found in whole food diets. Single high-dose antioxidant supplements can be problematic because oxidants have beneficial effects in low concentration and cause damage only when unchecked. Light-independent processes also produce reactive oxygen species, particularly under stress conditions, and require protection from xanthophylls. These stresses include pathogen attack or chronic psychological stress in humans, which can escalate to uncontrolled immune system attack on healthy organs. This is a root cause for a plethora of inflammatory diseases, disorders, and dysfunctional states. Dietary carotenoids keep a check on inflammation. Carotenoid-derived vitamin A has a well-documented immunoregulatory role, and a similar role is emerging for xanthophylls. Lutein and zeaxanthin also have roles in the prevention of neuroinflammation and thereby enhance cognitive function across the human lifespan, including attention, memory, learning, and executive functions necessary for complex tasks. Zeaxanthin protects against damage by intense light in plants and humans. The yellow center of the eye receives the brightest light and has the highest ratio of zeaxanthin to lutein. This preferential placement of zeaxanthin indicates a unique role in supporting vision in bright light. The enrichment of lutein in peripheral retina portions suggests a role in low-light vision. Zeaxanthin also shows an association with intense light in plants. Leaves rapidly form and remove zeaxanthin over the course of the day in sunny locations. Other leaf carotenoids of dietary interest, such as lutein and beta-carotene, do not change in concentration over the course of the day. Zeaxanthin plays a particular role in the harmless dissipation of potentially dangerous, unutilized light energy. The proportion of unusable absorbed light, zeaxanthin content, and energy dissipation rise and fall over the course of a sunny day. These dynamics are due to reactions that produce zeaxanthin and reconvert it to its precursor under limiting light conditions when dissipation of some absorbed light would be undesirable. Fast-growing edible crops utilize about 40% of midday sun for photosynthesis and growth and produce modest amounts of zeaxanthin. Slow-growing plants utilize much less of midday sun for growth and produce more zeaxanthin. Leaves of such plants are typically inedible. Ample zeaxanthin for human nutrition is thus hard to obtain from rapidly growing green produce. For improved human nutrition, we need crops that combine rapid growth and high zeaxanthin levels. Aquatic plants possess the unique ability to simultaneously grow very rapidly and accumulate exceptionally high zeaxanthin levels. This finding illustrates that both genetic factors and environmental factors can help maximize crop nutritional quality. Very high levels of zeaxanthin and related carotenoids are found in microorganisms living in extremely stressful environments, such as high levels of light or ionizing radiation, extreme heat, or extreme salinity. These microorganisms include the ancient ancestors of multicellular organisms like plants and humans. Archaeal microorganisms with extreme tolerance of heat and/or salinity contain the zeaxanthin derivative thermozeaxanthin. A bacterium that can grow on radioactive waste accumulates zeaxanthin and deinoxanthin. Ancestral functions of zeaxanthin and related xanthophylls are seen in microorganisms living in stressful environments. Zeaxanthin and lutein have wide-ranging protective roles in multicellular organisms. These functions intervene in oxidation cascades from formation of reactive oxidants to the regulation of key gene networks.